Okay, so these are related to enzymes X and Y. So one is substrate or the product or the enzyme. Okay, now you have to think. X and Y are the reactants, mean these are the substrates. In a chemical reaction where E is the enzyme, the product is Z. The first three stages in the reaction are given below. X plus E, first of all, X bind to the enzyme, then Y bind to that enzyme, okay, and they form a product. What would be the fourth stage? Okay, listen. What would be the last stage? What would would be the last stage? Now the product is formed. Exactly. It would get, sorry, क्या होगा? It would be D because product would get, wait. The reaction has occurred. Now this has finished, this all finished. Exactly, product would get detached, so it would be D. Enzyme would get separated from it because E is complementary to X and Y, not Z. Z is the product, it's a different thing. It's not complementary to E. Okay, Z is the product. So we have made the product. What would be the fate of product? The product is going to get detached. It's going to get separated from the enzyme. So answer would be D. They would get separated. Okay, amylase solution is tested with Benedict solution. Okay, now what is the function of amylase? To convert starch into glucose. It means your solution would have glucose, not starch, because you have used amylase. Okay, now we have tested it with Benedict solution, biuret solution, and iodine solution. What colors are obtained? Benedict will show positive, right? It is like red or brown precipitates. Okay, first of all, answer me. First of all, answer me. What is the end result of Benedict? That's the point. So, Benedict is used to test glucose or reducing sugar, and the end result is brick red precipitates. Okay, then biuret is used for the testing of proteins, and the end color is lilac or purple. Okay, iodine is used to test starch, and the end color is blue black. Okay, what is their original color? What is the original color of Benedict solution? Blue. What is the original color of biuret? Blue. What is the original color of iodine? It's brown. Okay, these are the original colors, and you should know these. Okay, so which which test would be positive and which would be negative? Would be positive and which would be negative? Okay, it would show test for only glucose, not the other one. So what is your answer? D. Yes, it would have a protein amylase. It's a protein. It would have glucose. Exactly. So purple color is shown because of amylase and this color is shown because of glucose. No, better listen. Amylase is present in the solution. Protein is present. Amylase is an enzyme. Enzyme is a protein. So protein will show a purple color, right? Only iodine is not going to give color because starch is decomposed into glucose. Yes. Oh, sorry. It would be C. Thank you for mentioning this. It would be C. Wait. Oh, why it should be? should it should be changed, right? Now this one is confusing for me as well. Okay, wait. Let me just, what colors are obtained? Benedict is definitely going to show color. This is also going to show color. That is for very sure. This one is the correct one. Okay, we can also say maybe some of the starch is present in the solution. So this brown color, blue black color would be shown. Okay, this can happen. वो तो definitely होगी और प्रोटीन भी definitely होगा. So इसके अलावा और कोई ऑप्शन नहीं है. Then we have to choose blue black. ठीक है. Next one. Okay, next one. इससे भी ज़्यादा मुश्किल है. आपने वाकई कभी ऐसे क्वेश्चंस नहीं किए? नहीं किए. These are very common. इसलिए तो मैं आपको खुद करवा रही थी. हाँ, ये तो बल्कि पूछे जाते हैं. उनको तो वैसे ही शौक है पकड़ के हमें करवाने का चीज़ें इस तरह की, जहाँ से बच्चों के नंबर चले जाएं. वो नहीं चाहते हम पास हों. Okay. A dish is filled with agar jelly containing starch. Okay. This all has starch. Starch, 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 starch. Now, four holes are cut in the jelly and each hole is filled. After 30 minutes, which hole will be surrounded by the largest area without starch? Now, amylase solution is going to decompose starch. What is the function of amylase? Starch into glucose. Okay. Now, what is the optimum pH of amylase? 7. Good. Now look at the B option. It has amylase solution, but along with that it has HCl. So it means it is denatured. Okay. Again, protease, we don't need protease for starch. For protease we need amylase, right? And the last solution is again protease, so we don't need this. So they are asking which solution is without starch. Amylase definitely. All other three would have starch because amylase is not there for working. Okay. Now some organisms live at the bottom of the sea where it is very dark. To answer is A. Exactly. Exactly. It breaks down starch. Okay. Some organisms live at the bottom of the sea where it's very dark. To synthesize glucose, they use energy from the chemicals in a very hot water that comes out of the volcanoes. What is a distinguished feature of these organisms? Their enzymes are easily denatured by heat. No, because they are present in a dark. Look at this one. They do not need to be green because there is no light. Okay, listen. They have already told you there is very hot water. They get energy from there. It means they need high temperature. They are not going to be denatured by the temperature. Every organism has a different optimum temperature. We have a different optimum temperature than plants. We have a different optimum temperature than birds. Okay? So here the optimum temperature is different for this species, maybe very high. So because they are in dark, so they may not need green color. Okay, next one is again a bit confusing. Now there are four test tubes given. Sorry, five test tubes are given with different pH. Okay, let me just copy it. किसको डांट पड़ रही है घर में? Okay, okay, okay. So here we have 2 pH, 5 pH, 8 pH, 11 pH and 14 pH. Now, just tell me, where in your body protease work? Protease, yes, protease work. Where is protease present? Is written from stomach. What is the stomach pH? 2. So what do you think, where would be the optimum activity or the maximum activity? 2. Okay. Let's say here would be the maximum activity. Let's go to the question. 5 tubes containing cooked egg white are set up as shown. They are cooked. Protease solution of different pH are added to the test tube. Which diagram shows the result of experiment for a protease from the stomach? Wait, wait, let's say in 14 it would be undigested. Undigested, right? 11 is also denatured one. 8 is also denatured one. These all are denatured. Yes, yes. The answer is C. So the digestion would take place only in A and B. So sorry, the 2 and 5. 2 would be, there would be maximum because the this one is optimum one. This one is okay, but it won't denature. Okay, next question. Okay, next the question number 7 is very, must be very easy for you. Okay, what is the enzyme that controls? Okay, 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 okay. Is it clear now? What is the enzyme that controls a reaction in both the enzyme and the substrate can denature at high temperature? That controls a reaction. There is one enzyme. There is one enzyme. Insulin is not an enzyme. To what? Okay. So amylase is 7 and lipase is 8.5. Okay. Okay, listen. We have a solution. We have a solution in which we have enzyme and we have substrate. Okay. Now there is a possibility that if we give this high temperature, it would get denatured. Okay. Now what should be done? What should be done to control this reaction? We have to use an additional reaction. Okay. So which enzyme should we use? 8 around 8.5. Hmm. Hmm. Look, look. You are comparing it with pH. I'm saying don't compare it with pH. Just tell me any enzyme that can control this one. So it's protease. It's protease actually. It's actually protease, but the reason that you're giving is not correct. Okay, so we don't have to consider it like what with respect to pH because they have mentioned in the question it's temperature. No, 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 that isn't correct. Don't have to mention pH. Okay, wait. Now we have put a solution into a beaker. Now we have to use something additional. Okay. So they're asking what is the enzyme that controls a reaction in which the enzyme and substrate can denature at high temperatures? Okay. Both can denature. So among these, only proteins can denature. So if if protease is present, it must be acting on proteins. No, 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 because in saliva there is no protein. Exactly. Again, no, 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 no. They won't denature. They won't denatured. Okay. Only proteins can be denatured. Okay. Last one. The diagrams. No, yeah, we have time. The diagram represent an enzyme and four molecules A, B, C, D. Which one is the perfect one for this? Look at the shapes. Molecule C. Correct. Okay, let's discuss the next three, and then you have to do the rest by yourself. And then we'll discuss those tomorrow. But please, you have to attempt these or go through these. If you don't want to attempt, it's okay. Just go through this document. Okay. Okay. The question is a bacterium lives in a hot spring. Now, the optimum temperature is 85. Okay. No, the activity is maximum at optimum temperature. Exactly. Okay, which graph represents that enzyme? Where D? Okay, D. Okay, next one is the ninth one, and then the 10th one. Okay, ninth one is quite easy. It's related to the diagram. You have to tell the correct diagram. Okay, okay. We have a key, lock and key model. There is an enzyme and there is a substrate which can easily fit into it. Now, this works well at pH 7. Okay. Now we have changed the pH. It must get denatured. Means the shape changes. So which shape would it be? Only the shape of exactly. Only the shape of enzyme would change. The substrate would remain same. Okay. Now the last one, the 10th one. The diagram represents how an enzyme molecule changes its shape. Again, it's related to shape. And then what explains the change? Why it's changing its shape? Good. 2, 70 correct. Okay. So this is all for today. You have to go through